This video is on VIRAGEN real-time RTPCR master mix preparation and programming. The VIRAGEN kit contains the following components. First is a lyophilized enzyme mix, primer probe mix, a positive control, a negative control, RNase-free water, and an enzyme mix buffer for the resuspension of the lyophilized enzyme mix. And finally, a user manual. Please follow good laboratory practices. Clean the lab surface before starting the reaction. First step is the PCR master mix preparation. Add 400 microliter of PCR buffer and 500 microliter RNase-free water into the lyophilized enzyme mix provided in the kit. Pipette slowly. Avoid bubble formation. Later vortex lightly. After the resuspension of the lyophilized enzyme mix, we can proceed for the PCR reaction mixture preparation. For easy understanding, I will explain here the reaction volume for one reaction. According to the number of reaction you are planning to keep, multiply accordingly. Take a fresh append-off. For a single reaction, add 9 microliter of resuspended enzyme master mix to the append-off. Next is the addition of primer probe mix. Add 1 microliter into the master mix. Since we are handling with small volume, pipette slowly and carefully. Do not add to the walls. Add directly into the mix. Next is the addition of 5 microliter RNase-free water to make up the volume into 15 microliter. Rest 5 microliter will be the sample. Total volume of the reaction will be 20 microliter. If you are planning to use 10 microliter of sample, avoid the addition of RNase-free water. Instead, directly add 10 microliter of sample. Aliquot the reaction mixture into PCR strips, tube or plate which is compatible with the machine. Ideally, minimum of 3 reactions are required in a run. The sample you are planning to test, 1 positive control and 1 negative control. So aliquot 15 microliter to each PCR tube. Mix properly and then add. Here we are running multiple samples. So according to the number of reactions you are planning to do, prepare the master mix and then aliquot accordingly. And it is always recommended to prepare 1 reaction extra to avoid the pipetting error. Add 5 microliter of negative control into a tube and close it before taking it for sample addition. This is to avoid cross-contamination with other samples and positive control. Next, take the tubes to the dedicated space for sample addition. Add 5 microliter of samples to each tube. The samples you are adding is the RNA extracted from the patient collected samples. Extraction of RNA is a crucial method before the PCR reaction. Finally, add 5 microliter of positive control into the dedicated tube. Spin down the tubes to collect all the samples to the bottom of the PCR tube. Next, you can set up the program. In this demo, we are using Bio-Rad CFX96 real-time machine. After opening CFX Mastro software, you can click on New Protocol to enter the program. The program is as follows: 50 degree 10 minutes, 95 degree 3 minutes, next is a 40 cycle run, 95 degree 15 seconds, 55 degree 40 seconds. Set it for 40 cycles. And at last 25 degree 10 seconds. Finally, you can change the volume to 20 microliter and then save the program. Next is the plate layout. Click on wells in which you are planning to keep the tubes. First, select the fluorophores. For VIRAGEN kit, select FAM, hex and rocks. Then you can select the sample type. Enter positive control, negative control and unknown sample. After clicking all the fluorophores, you can enter the sample name if you want. Finally, save the plate layout. Now you can open the lid of the machine and keep the tubes as per you have saved the plate layout. After closing the lid, you can click on Start Run. Save the program and the program will be initiated.